Thank you for the introduction, and thank you very much for inviting me as a speaker. So I think I did a presentation in this BrainMap seminar in 2012 when I was a postdoc, and I'm very happy to be here again. And after my postdoc for about 10 years, my research interest and goal have been focused in the advanced fetal brain MRI processing and analysis for providing and developing imaging biomarker of early brain development. So today I will present our several studies about method development and also normal and abnormal fetal brain MRI data analysis and introduce some significant research findings. First, human brain shows very dynamic developmental process and major neurogenic event, including neurogenesis and neuronal proliferation, migration, and differentiation. And cortical neurons originate in the ventricular zone and subventricular zone and migrate to their proper lamina and aerial position of the cortical plane. And then their neuronal connectivity and thalamocortical axonal development and the association cortical fiber development also start during the fetal stage. And along with this connectivity development, we can see very rapid brain size growth and very, very fast cortical gyrification from smooth to highly folded structure. So that folded structure is fully determined before birth. And this human brain, fetal brain developmental process is largely controlled by genetics and then largely influenced by several maternal and environmental factors such as maternal socioeconomic status and maternal nutrition, mental health, and maternal obesity, and also closely associated with the placental oxygen transport to the brain and other organ development. And if there are any negative effect on the brain development, that brain structure and function become atypical and abnormal, and it will finally affect lifelong neurodevelopmental and cognitive outcomes. So to understand this relationship and this effect, and then to understand all these mechanisms, in utero in vivo fetal MRI must be very useful modality to understand that. So compared to ultrasound, fetal MRI can provide better soft tissue contrast and various structural and functional information. And among fetal cases showing any brain abnormality detected in ultrasound examination, fetal MRI can provide additional diagnostic information in 50% of those cases, and it can change prognostic information in 20% and clinical management in more than 1/3 of the cases. But even with the fetal MRI, visual assessment, the error rate of visual fetal MRI diagnosis is around 7-8%, and it's very challenging to determine whether that brain is normal or abnormal because fetal brain development quantification is not very perfect yet. So we often miss subtle brain abnormalities during the fetal stage, and it is even more challenging to predict future outcomes. So visual fetal MRI assessment, it shows low predictive power for future poor outcomes. So it's only 41-48%. And the outcome prediction quantitative MRI technique is very, very limited. So our goal is advanced and accurate quantitative analysis of fetal brain development to better understand human brain development and to identify those effects and develop imaging biomarker to characterize typical or atypical brain development and also to predict future brain abnormalities and future cognitive neurodevelopmental outcome. But to achieve that goal, we have two approaches. The first one is a surface-based regional tissue compartment analysis. So still, many fetal MRI studies, they are using only volumetric measures and they are analyzing some structural measures at a whole brain global level. So this approach aims for more sophisticated, more detailed, and more regional structural analysis. So to do that, we developed the fetal brain MRI processing pipeline, and based on that processing pipeline, we have been analyzing fetal cortical plate and subplate regional growth in typical fetuses and fetuses with several brain developmental disorders. And another my long-standing research topic is graph-based sulcal pattern analysis. And recently, we advanced that method by using AI method. And another approach is deep learning-based fetal brain MRI analysis.
Vino un pic, vrei să vezi o prezentare cu mine?
Can I go on? So in this approach, we aim to extract meaningful feature and structural pattern and any brain abnormality from nearly raw format MRI data. So with the minimal data processing, it can reduce and minimize non-biological technical variability. So I will talk about our method of fetal brain age prediction and unsupervised fetal brain abnormality detection. So these two approaches are complementary to each other. First, surface-based analysis can give us scientific findings with more clear biological meaning based on specific hypothesis, but we need a complex data processing with many steps, and it often requires manual editing for reliable data processing. But deep learning-based analysis, we don't need a manual editing for that deep learning-based analysis, so it could be fast and real-time for new subject analysis, and also maybe in the future it may enable more practical clinical use in the future, and also it can reveal some hidden, unknown pattern of normal and abnormal brain development from imaging data. However, result interpretability is still limited. So I will talk about these topics one by one. So first, I will quickly talk about fetal MRI. Compared to postnatal brain MRI, the biggest issue in fetal MRI is unpredictable extreme fetal movement. So fast single-shot tip-weighted imaging is performed for structural imaging. So in-plane imaging shows good structure, good tissue contrast, and is less susceptible to fetal motions. But there are interslice motions and misalignment between slices. So fetal MRI protocol usually acquire multi-view, multiple steps to see brain anatomy more clearly in different views. So because of this different imaging characteristics, image processing pipeline specialized for fetal brain MRI is necessary. So we'll talk about our fetal brain MRI processing pipeline. So this is the whole entire fetal MRI processing pipeline from multiple raw MRI stacks to the surface extraction and the regional postulation. And in this pipeline, one of the most important part is motion correction and super resolution volume reconstruction from these multiple raw MRI stacks. So for reliable volume reconstruction, accurate brain masking, and the exclusion of a bad quality MRI stack, doing that thing before reconstruction is preferred because in our experience, if we don't exclude non-brain tissue region in that MRI stack, and if we don't exclude bad quality MRI stack, it definitely lower the performance and the quality of 3D volume reconstruction. So with high quality brain extracted multiple input image data set, input image stacks, we reconstruct the motion corrected 3D volume using sliced volume registration or deep neural network based sliced volume reconstruction. And we segment cortical plate and subplate tissue regions using our attention unit model with the aggregation of multi-view predictions and test time augmentation. So this is the example. Okay. Yep. For this quality assessment, just 3D input data. So we use the 3D CNN model.
Yeah, but get a sense for, I see that there's not many slices there. do you know contributes to the metric?
Yeah, so image noise and the motion level. So usually bad signal and then severe motion, yeah, that affect the score. So we manually score, yeah, that thing from 0 to 1, 0, 0, 0.25, 0.5, and 5 level score. So that's a training data set. And then we train the 3D CNN model. And then, yeah, because this raw MRI stack, if it shows bad signal or if there are very serious interslice motion, it affects a volume reconstruction performance. So, yeah. Okay, so these are the example for automatic cortical plate and subplate segmentation across different gestational ages. And in our test, the segmentation accuracy was higher than 90%. And from the segmentation images, we reconstruct 3 surface model. So inner cortical surface, outer cortical surface, and inner subplate surface. So first, we reconstruct the inner cortical surface, and for outer cortical reconstruction, we deform inner cortical surface outward to fit the boundary between cortical plate and CSF region. And to do that, we first define deep sulcal CSF region using a modified skeletonization algorithm, and surface deformation is guided by a Laplacian gradient map. For inner subplate surface model, we deform inner cortical surface in a different way. So deform the surface inward to capture the boundary between subplate and intermediate zone. And in this surface deformation process, we empirically determine different parameter weight set for different gestational age, because fetal brains, they have a very highly variable brain size and cortical gyrification. So we need a different parameter weight set for different age. So as a result, this tool accurately captured the boundary between different tissues at any gestational age and make the surface model smooth and topologically correct without mesh self-intersection. And after cortical and subplate surface extraction, we are doing automatic sulcal labeling. So we developed the novel sulcal labeling method for the fetal brain. So to do that, first we generate sulcal probability maps on the multiple template brain with different age. And for individual brain sulcal labeling, weighted sulcal probability map from multiple templates are defined by measuring gyrification similarity between individual brain and template brain. So for example, for that individual 27.5-week individual brain, sulcal probability map of 28-week template will be more weighted. And then that weighted sulcal probability map will be registered to the individual brain by using surface registration. And then we assign anatomical sulcal label to each sulcal fold. So the mean accuracy for this sulcal labeling was higher than 95%. And more recently, we also developed the fetal cortical gyral pastillation using deep learning. So we developed the attention-gated spherical unit model for fetal cortical gyral pastillation. And this model takes average convexity sulcal depth and mean curvature feature maps, and the output is a pastillation label. And our model showed higher accuracy in the cortical gyral pastillation compared to other methods, including surface registration, original spherical unit, and SPROMNet. So with the process surface data with the regionally pastellated surface data, we are analyzing cortical plate and subplate growth and sulcal folding patterns. So I will talk about the normal, abnormal brain MRI analysis. And we have published several studies about normal typical pattern of regional cortical surface growth and folding. And in this study, we identify spatial temporal pattern and variability of early sulcal folding. And we identify this hemispheric asymmetry and sex difference. And more recently, the onset of cortical folding in fetal brain was correlated with the cortical plate microstructure by combining with the diffusion MRI analysis. So in that study, we used the public developing human connectome project data. So we used the diffusion structural fetal MRI data of 112 subjects, and we mapped the sulcal depth value and cortical plate microstructural diffusivity values on the inner cortical surface model. And the tissue fraction and fluid fraction value were mapped along the normal vector on the cortical surface model. And here, increase in the cortical plate tissue fraction may reflect the increase in cellular density and the synaptic density and dendritic vaporization. So after mapping folding information and the microstructural diffusivity value on the same surface model, we did the coupling analysis within a subject. So we define a small patch region on the surface, and on that region, we examine the correlation between tissue fraction and sulcal depth. And we found a very strong negative correlation between these two factors in most of the cortical regions. So it means a deep sulcal region has a lower tissue fraction. And in addition, we also found a relationship between future cortical folding map and tissue fraction. So we projected future cortical folding map from all the fetal brain template onto the individual brain using surface registration. And we did the same coupling analysis. And then we also found in many cortical regions, we found a negative correlation. So this interesting results showed that on the unfolded smooth region, tissue fraction values were already lower in the areas that develop into deep sulcal region in the future. So we confirmed that from this study, we confirmed that neuroanatomical difference between sulcal and gyral regions already exists in fetal life. And in our study, our hypothesis is that high tissue fraction may reflect high neuronal density in that region. So neurons more accumulated in that region. And then that high tissue fraction areas show more cortical expansion and greater cortical tangential expansion and develop gyral folding later. So this result can give us one more clue about cortical folding hypothesis. And we also detected locally abnormal cortical surface area and folding growth in abnormal groups. So in fetuses with Down syndrome and in opioid-exposed fetuses, we showed the abnormal cortical surface area and folding growth. So this study showed that alteration in cortical growth in this abnormal group is already region-specific before birth during fetal life. And also, it is very important to study heart, placenta, and brain triad to understand human brain development, because heart, placenta, brain, they are highly interconnected and they have a hemodynamic interdependence during development. So we studied the effect of a placental oxygen transport and the effect of a congenital heart disease on fetal cortical surface growth. So I will introduce these two publications. So to study the effect of placental oxygen transport by collaborating with Ezra Bachtar in our center, we did the twin studies. So because of twin fetal brain, they have the identical genotype under the same maternal effect. So we can look into the placental effect, only placental effect after controlling for other vectors. So for twin brains, we perform placental bold MRI with the maternal hyperoxygenation, and we observed the signal change during hyperoxia. And then we measured time to plateau values, indicating the efficiency and speed of oxygen transport. So we measured TTP value for each twin, and also we measured regional cortical surface metrics for 2 twin brains. And our results showed that twin fetuses, one fetus with a high TTP value. So fetuses with the slower and less efficient oxygen transport, that brain showed reduced cortical surface area growth in the frontal lobe even after correcting for whole body size and whole brain size. And then, additionally, TTP value difference and global circle folding pattern similarity was negatively correlated. So it means a greater discordance in oxygen transport level may result in the greater difference in brain structure between twin brains. And another study is about congenital heart disease. Human brain development is significantly affected and impaired by congenital heart disease because of the genetic effect and altered blood supply to the brain. So in this study, we analyzed the cortical surface metrics for automatically postulated cortical regions from longitudinal MRI data and found the significantly reduced regional cortical surface area growth in fetuses with congenital heart disease after 30 gestational weeks compared to control fetal subject. And the more notable and interesting result is that lower regional cortical surface area growth was significantly correlated with the worst 2-year neurodevelopmental outcomes. So more deviation of a brain structure in utero lead to worse cognitive and motor language outcome score. So this is the first study to find the significant relationship between prenatal regional cortical growth and postnatal neurodevelopmental outcome. So we think prenatal cortical growth, surface growth, can be a critical feature for predicting future neurodevelopment. And we are interested in not only surface area and folding, but also cortical plate and subplate thickness. And recently we have been focusing on subplate thickness analysis. Subplate region is in the fetal brain temporary transient compartment just beneath the cortical plate. And that region is well known as a waiting area for thalamocortical and corticocortical fiber development. So the hypothesis is that subplate thickness could be a marker of the amount of cortical connections. So our surface models, 3 surface models, inner subplate surface and outer subplate surface and outer cortical surface, they have the vertex correspondence to each other. So subplate thickness can be estimated by computing the Euclidean distance between linked vertices of the inner and outer subplate surface models. So these are the maps of mean subplate thickness for each age range every 2 weeks, and then global whole brain mean subplate thickness increased with the gestational age. So we can see regional variability. So we analyze that regional variability more deeply. In 68 typical fetuses ranging 22 to 32 gestational week, we identified spatial variability of a subplate thickness after correcting for confounders such as gestational age and sex, and parietal region and perisylvian region showed greater thickness than other region. And when we compare the cortical thickness between the second trimester and third trimester, each region shows different relative thickness change between these two time periods. And we observe the thickness growth trajectories, and we calculate the growth rate at each time point, and we cluster the regions having a similar pattern of growth rate changes over time. Then it shows the cluster regions of subplate thickness growth and surface growth. And here, subplate thickness shows one more cluster and showed more heterogeneous growth pattern across the regions and across the cluster compared to surface growth pattern. So this regionally variable subplate thickness growth might be associated with the spatial and temporal variability of thalamocortical and corticocortical input and connectivity density. And we also found significant hemispheric asymmetry in subplate thickness. So lateral frontal region, post-central, and the medial temporal region showed rightward asymmetry, and parietal and medial frontal region shows significant leftward asymmetry. So as you know, brain structural and functional asymmetries are very well-known feature in the human brain, and we demonstrated that brain asymmetry are already present in the subplate structure in early fetal stage during the second trimester. So I think this study revealed the earliest observed symmetry in the human brain ever, as far as I know. Yep. This is cross-sectional data. Yep. Okay, so on the cortical surface, another biological important feature that I've been studying for a long time is the global sulcal folding pattern. So the sulcal patterns are formed by the arrangement, number, size, and depth of sulcal fold and their complex geometric and topological relationships. And this primary sulcal folding pattern are spatially prenatally determined, and then those patterns are relatively stable throughout life. And then that sulcal and gyral folding pattern are spatially correlate with the early gene expression in the brain, and also that patterns are thought to be associated with the cortical functional arealization and the cortical fiber development. So sulcal pattern has a very critical information about early brain development related to brain function. So my original idea was to use a graph structure based on sulcal pit as a node to characterize and represent and analyze complex sulcal folding patterns. So a long time ago, I developed a graph-based quantitative sulcal pattern comparison and analysis method. And the sulcal pattern graph has the geometric information of sulcal fold, such as 3D position, depth of the sulcal pit, and sulcal basin area. And that graph also includes the internode, intersulcal geometric relationship, and sulcal graph topological features to characterize the arrangement and patterning of sulcal fold. And this sulcal pattern graph can be optimally matched and compared with other brain sulcal graph using spectral-based method, and we can measure sulcal pattern similarity between these two brains ranging from 0 to 1. Yeah?
How do you sulcal connections between the nodes of the graph?
Yeah, if the sulcal basin meet geometrically on the surface, it will become edgy. So it's not about the fiber diffusion connectivity. Yeah. Yep. So this method has been validated through many application studies, and several patient groups shows low sulcal pattern similarity to the normal control group. And we applied this method to fetal brain. So the approach is to compare individual circle pattern graph to the circle pattern graph of the fetal normal brain template. So we measured the circle pattern similarity to the normal template for individual fetal brains. So here we found from many studies, we found a lower, significantly low circle pattern similarity to the normal template compared to typically developing fetuses, such as ventricular megaly or fetuses with congenital heart disease and agenesis of corpus callosum, and fetuses with the brain malformation, polymicrogyria, and child 2 malformation. So all these studies showed high sensitivity in detecting abnormal cortical structure compared to simple folding measures such as the gyrification index. For example, in this brain malformation study, one brain was initially judged to have a normal folding by visual MRI assessment, but our quantitative circle pattern value was very, very low, and it was within lower 5% of the distribution of the typical subject circle pattern values. And when we checked the follow-up neonatal imaging, that brain was finally confirmed to have a abnormal cortical folding and have a perisylvian polymicrogyria. So qualitative fetal MRI assessment missed subtle folding abnormality, but our circle pattern method detected that thing in early fetal stage. Yep. So yeah, we can get the pattern similarity values. So we use those values, and then it's for the global now. So if you want a regional analysis, we need to define those areas. Yeah, but I think just analyzing only one sulcus is not very meaningful, because one sulcus, they have only 2 or 3 folding segments. So it's more focused on the global pattern of the sulcal folding, like looking at the fingerprint. Yeah. And in our most recent study published this year, we also revealed the significant relationship between fetal sulcal pattern abnormality and 2 years developmental outcome in subject with ventricular megaly. The ventricular megaly patient, they showed large ventricle even in the fetal stage, and some ventricular megaly patient, they show impaired neurodevelopmental outcome, and they show developmental delay, but some other patients do not show any problem. Their cognitive outcome and developmental outcome are all fine, but there have been no clear marker to predict their future developmental outcome. But in this study, the subject with the delayed developmental outcome, they have more atypical fetal sulcal pattern compared to the subject with age-appropriate outcome. So this study shows the potential of fetal sulcal pattern features as an early image marker to predict postnatal neurodevelopmental outcome. But there are some methodological limitations, because for similarity value calculation, we assume equal contribution of all sulcal folding regions, and for regional analysis, we need a predefined area. And to calculate sulcal pattern values, we are using, as I said, position, depth, area, and graph topological information, and we need the feature weight. So those feature weights are empirically determined by users, so it may not be very optimal, and the spectral graph matching method is computationally very complex and intensive, so it takes a time to calculate the sulcal pattern feature for a new subject. So to overcome these limitations and to advance our method, essentially we developed a prototype-based graph neural network for sulcal pattern analysis. So this is supervised learning to classify the group or predict something, and then this model learn and identify the prototype of circle pattern for each class. And then for a new tested subject, this model measured the similarity to a set of predefined circle pattern prototypes. And based on that similarity values, we performed the group classification for the patient and control group. And this model was trained with the control and congenital heart disease patient circle pattern graph data, and this model showed the higher accuracy in classification performance compared to the other state-of-the-art models. But this model is not yet designed for fetal brain, so our plan is to expand this method for fetal circle pattern analysis. Okay, so now let's move on to the deep learning-based fetal brain MRI analysis. I will talk about fetal brain age prediction first. And brain age is the biological age of the brain, and I think maybe you already know about that thing. The brain age gap between the estimated brain age and actual chronological age can be a meaningful biological marker for characterizing typical brain development, aging, and brain disorder. So in the MRI-based machine learning method for age prediction, we can train a model with MRI and age data, and for new tested subject, using MRI data, we can predict their brain age, and we can measure brain age gap, and that brain age gap value will be analyzed. I'm sorry, what age range of the? Yeah, so your question was the age range for that brain age prediction. And previous studies, there is a lifespan study, so they are predicting brain age from 0 to 90 years old. So they are using all publicly available data sets. So using more than 100,000, something like that. But in my study, I'm focused on fetal brain MRI. So just we are predicting from 20 weeks to 40 weeks. So the error value was the kind of 2 day or 3 days. Yeah. So yeah, I'll show the detail of the method and result. So yeah, we can predict fetal brain age using MRI data as other studies are doing. But we need to think about the optimal approaches for the data input and the model structure, because fetal MRI has a unique characteristics. So when you see raw fetal MRI stack, in-plane imaging is good and it shows brain structure very well. But in other views, there are interslice motion, and the slice thickness is between 2 and 3 millimeter. So in other view, brain structure anatomy doesn't look clear at all. So I think the 3D whole MRI stack as an input is not very helpful for accurate fetal brain age prediction. So some studies, they are using only one center slice of the brain, 2D slice, but using single slice is a little risky, because when you see in-plane slices, some slice have low quality and shows a bad signal, and then they have a lot of noise. So using one single slice is risky, because it may choose a bad quality slice by chance. So just we select 4 to 6 slices near the center of the brain, and we predict brain age for each slice using 2D single channel CNN model. And then for more stable output, we perform test time augmentation. So we aggregate the multiple brain age values, the prediction, and then we take a representative value, such as the mean, median, or mode of multiple age prediction. So this approach is more robust to interslice motion and slice selection and any outlier or error values. And mean absolute error in age prediction was 0.2325 weeks. And our 2D single channel model with the aggregation of the multiple predictions showed better performance in age prediction compared to the multi-channel or 3D model. So we applied this method and analyzed fetuses with ventricular megaly. So we predicted fetal brain age for typically developing fetuses and ventricular megaly fetuses. And we compared brain age gap values between these two groups and also among subtypes of patient groups. So mild, moderate, and severe ventricular megaly cases were compared, and also isolated ventricular megaly and ventricular megaly with other additional brain abnormality. Those groups were also compared. So first, ventricular megaly fetuses showed significantly larger brain age gap value than typical fetuses. And we also found larger brain age gap in severe ventricular megaly fetuses with other additional brain abnormalities compared to mild cases with isolated ventricular megaly. So this study shows that fetal brain age prediction has the potential to be another marker to assess fetal brain health and abnormalities. But brain age is a global measure of the whole brain, and it cannot detect any regional brain abnormalities and alterations. So this is an ongoing project. So we aim to measure local fetal brain age at a voxel level, and this shows the local brain age map and local brain age difference map. And now we're working on improving and optimizing the method, and we plan to apply this method to abnormal fetal brain MRI, and we want to validate whether it's useful for patient analysis, and we want to validate whether it's useful for regional abnormality detections. Yep. Sorry? So you're asking about the validation approach for local brain age. I think local brain age gap value can tell us the performance of the local brain age estimation. It's the same with the global brain age estimation. So always the accuracy for predicting brain age for normal subject data. The brain age gap value during the model training and for the test data, I think if we acquire lower brain age gap value, it means that model is working well. And of course, we need to make that model more generalizable. So we have to do some external validation, and then we have to apply that method to the other various data sets. But usually for normal subject, brain age gap value, if we have a lower mean absolute error value, it means that that model is working well. Okay, and this is the last thing. So I will present our deep learning-based normative modeling and anomaly detection of fetal brain development. So in a conventional normative modeling, approaches to extract specific brain features such as the brain volume, head circumference, and the whole brain cortical surface area from large normal population, and we create the model using statistical methods such as very well-known generalized additive model of location, scale, and shape, Gaussian process regression, and hierarchical Bayesian regression. And then for individual subject, we compared that individual subject with that normal distribution, and we measured the z-score or percentile score. But deep generative reconstruction-based normative model can learn normative pattern from imaging data itself during the training if we train this image reconstruction using normative subjects. So after training the model, when abnormal brain MRI data is given to that model cannot reconstruct original data well because that model was trained only with the normative subject. So that difference map between input data and generated MR imaging data, that difference map can be considered a brain anomaly map. So, and then usually variational autoencoder and generative adversarial network and diffusion model have been used for this purpose. So in our study as a baseline, we implemented and tested the backend model. The backend model showed low performance in fetal brain MRI reconstruction, and the reconstructed MR images are very, very blurred. So I think one of the main reasons for this is very high temporal variability and highly variable brain size and folding structure of fetal brains. So this basic model shows difficulty in reconstructing fetal brain images. So we developed and proposed a conditional cyclic backend model. So we call it CC backend model. And a key feature in this model was to add a brain age estimation module for age conditioning. So this model can capture temporal variability of fetal brain structure and reconstruct age condition output MRI. So our backend model showed much better performance in reconstructing fetal brain MRI. So when compared to input original imaging, generated image shows a very similar, clear imaging. So the CC backend model showed the lower mean absolute error and mean squared error between input and generated images compared to a baseline backend model. And the whole brain mean absolute error between input and generated images, that value was used as the anomaly score. So we measured whole brain anomaly score for typical fetuses and fetuses with the brain malformation, several brain malformation, including ventriculomegaly, polymicrogyria, having weird folding, and microcephaly, very small brain, and macrocephaly, very big abnormal fetal brain. And then those brain malformation cases showed the much higher anomaly scores compared to typical fetuses, and the value anomaly score distribution of a brain malformation totally deviated from the normative value distribution. So in a group classification, just it shows the perfect group classification. So AUR's value was higher than 0.99, which is almost perfect. So in addition, we did the separated site experiments. So here we completed separated data source between the test and training data set for external validation. So of course, classification performance become lower, but the still AUR's value was still high. It's around 0.85. And if we register this anomaly map to the regionally postulated fetal brain template, we can measure regional anomaly score for individual brains. So using this method. So for example, this case is a ventriculomegaly brain. So after running registration, and when you measure the regional anomaly score, that lateral left and right ventricle, that region showed high anomaly score. So using this method, we will be able to get a regional anomaly map and regional score from nearly raw format MRI data. And I think it might be helpful for radiologist fetal MRI assessment in the future. So our goal and challenges is we have to make our fetal MRI processing pipeline more generalizable, because now bad quality clinical MRI or abnormal brain MRI data showing very, very different brain structure, our processing pipeline sometimes does not work well for those MRI. So we increase the generalizability of the MRI processing pipeline with a multi-site large data set or synthetic data set. And of course, we always need the advanced methodology development and improvement for higher accuracy and reliability in data analysis. And also, longitudinal multimodal data acquisition analysis is a big direction to go. So to understand human brain development and also to develop any brain development model, we need to understand the brain developmental trajectories from fetal stage to infancy, childhood. And then brain MRI features need to be more correlated with the many other factors, genetic, maternal, environmental factors, and neurodevelopmental outcomes. And also AI-based individual patient prediction using all this multimodal data will be a very big future direction. And then we also want to integrate fetal brain structure, function, and connectivity using multimodal data. And we need to design a study to enhance and validate clinical utility of our method to advance precision medicine. Okay, so thank you very much.